Welcome to The Explainer. Today, we're diving into something that's totally revolutionizing science and medicine, CRISPR-Cas9. It's a gene editing tool that's, frankly, mind-blowing. And right off the bat, it's pronounced CRISPR.
Okay, let's just start here, because this quote really nails it. Think about the entire human genome, all 3 billion letters of our DNA, as one gigantic book. Well, CRISPR is basically the find and replace function for that book. It lets scientists zoom in on a single word or even a single letter and change it. It's that precise.
So here's what we're going to cover. First, we'll get into what CRISPR actually is. Then, we'll see how these little molecular scissors really work. After that, we'll look at what happens after the cut is made, which is super important. We'll also walk through how scientists plan and edit. And finally, we'll touch on the absolutely incredible future this technology is unlocking.
So, what is this thing? Where did this incredible genetic toolbox even come from? Well, you might be surprised to learn that scientists didn't just invent it out of thin air.
Okay, the name itself, CRISPR, is a bit of a mouthful. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now, don't worry about memorizing that. What's important is that it describes these weird, repeating patterns of DNA that were first spotted in bacteria. And those patterns, it turns out, were part of something really, really cool.
You see, this whole system is actually a tiny, ancient immune system for bacteria. Yeah, that's right. When a virus attacks a bacterium, the CRISPR system grabs a little piece of the virus's DNA and saves it. It's like taking a mugshot. So if that same virus ever shows up again, the bacterium recognizes it immediately and sends out an enzyme to just chop up the viral DNA. Threat neutralized.
And this is where scientists had their big aha moment. They realized they could reprogram this bacterial defense system. It really just comes down to two key parts. You've got the Cas9 protein. Think of this as a pair of tiny programmable scissors that can cut DNA. And then you have the guide RNA. This is the GPS. It's a molecule you can design to lead those Cas9 scissors to exactly the right spot in the entire genome.
So, you have your scissors and you have your GPS. How do they actually find their target and make that cut? Well, it's this incredibly elegant three-step process.
It all happens in this beautiful little sequence. First, the whole complex scans along the DNA, and it's looking for a very specific landing pad, a short sequence called a PAM. Once it locks onto that PAM, the guide RNA unwinds the DNA right there and checks to see if its own sequence matches the DNA sequence. If, and only if, it's a perfect match, the Cas9 protein changes its shape and snip, it cuts right through both strands of the DNA.
Now, making the cut is actually just the start. The really amazing part, the editing part, happens next. The cell's own natural repair crews rush in to fix the break. And it turns out the cell has two main ways it can patch things up.
And these two pathways are what scientists can take advantage of. On one hand, you have what's called NHEJ. Think of this as the fast and dirty, quick fix. The cell just jams the two broken ends back together. It's often messy and creates little errors, insertions or deletions called indels that usually just disable the gene. But on the other hand, you have HDR. This is the precise edit. If scientists provide a correct DNA template along with the CRISPR system, they can trick the cell into using that template to make a perfect repair, effectively replacing a faulty gene with a healthy one.
Okay, so we get the theory, but how does a scientist actually take this from an idea on a computer to a real experiment that works? It's a pretty careful and methodical process.
It all starts with a game plan. You've got to figure out first, how are we going to get this CRISPR machinery into the cells? Then, what's our goal here? Are we just trying to knock out a gene, or do we want to do a precise repair? From there, you design the perfect guide RNA for your target. You test it to make sure it works well, and then, after the experiment's over, you have to go back in and check the DNA to make sure you got the edit you wanted. And that very first step, delivery, is actually one of the biggest hurdles. You have a bunch of options. In a lab dish, you might use something simple like a plasmid, but to get it into a living animal or a person, scientists often use a harmless modified virus like a lentivirus or an AAV to act like a little delivery drone. Each method has its own trade-offs, when it comes to efficiency, safety, and just how much stuff you can pack inside.
So look, this is way more than just a cool lab trick. The power to literally rewrite the code of life, it has this almost unimaginable potential to change everything.
I mean, just try to wrap your head around the precision we're talking about here. Scientists can find and fix one single letter that's wrong out of the 3 billion letters in our entire genome.
That's like finding one typo in a library containing thousands of books and fixing it without messing up a single other word. It's just incredible.
And because it's so precise, it's opening the door to potentially curing diseases that, until now, have been a life sentence. We're talking about devastating genetic disorders like sickle cell anemia, Huntington's disease, and so many others. And this isn't science fiction. Clinical trials are happening right now, showing real, tangible hope for patients.
The CRISPR revolution is really just getting started. It's already transformed basic research, and it's poised to transform medicine and agriculture. What's been done so far is amazing, but it really feels like we're just scratching the surface. It leaves you wondering, what incredible breakthroughs will it build next?